Okay, so we are now on the Hangout live on the air, and we've got 30 more seconds, so I can just do a long rambling introduction of Dr. Maresca. So, very happy to have Julie giving us the talk today. She is in our Civil and Environmental Engineering department here at UD, and had done her postdoc with Ed DeLong at MIT, and before that was grad student with Don Bryant at Penn State. And so she is going to tell us today about the world without phosphorus and some microbial ecology and I think metagenomic stories from Indonesia, right? And hopefully some good field photos, right?
I have a few at the end. I wasn't sure how long the slides would take.
All right. Well, so just a reminder to everyone in the Hangout that you should shut your mics and video off as Julie is speaking, and to anyone watching, a reminder that you can tweet questions to MicroSeminar or use the hashtag #USeminar, and we will ask them on air after Julie has completed her talk. Okay? So with that, we can share screen, Julie, and have you get going.
Great. Well, thanks a lot for the invitation to give this talk. I've never given a seminar online before. It's kind of exciting. I feel like it's my TV debut.
It looks good.
Can you hear me okay?
Yep.
So what I'll be talking about is some work we've been doing with bacteria isolated from a phosphate-limited lake in Indonesia, and how they've managed to live with very, very limited phosphorus. The lake looks like this. This was the picture in my trailer as well, and this is a picture that I got from Sean Crow, who has been leading the fieldwork at this lake for many years now. And one of the really striking things about the field site when we got there was that the water is totally clear. You can swim around and see your toes. There's really nothing in it. And what that told us, even without doing any experiments, was that there was something that was limiting the growth of algae and other organisms that normally are the particulate matter in water that scatters and absorbs light. So we were wondering, among other things, what was limiting the growth of those microbes? And what was limiting the growth of those microbes and how they get around that. In actual fact, there are many ways to get around resource limitations, right? And we all know this from whenever gas prices get very expensive. You know, we diversify the resource acquisition so we can get fossil fuels from different sources than normal. We can substitute more abundant resources, so for example, biofuels. We can reduce our demand, so start biking or walking instead of driving. And of course, increase efficiency. Microbes use exactly the same set of strategies to grow when resources are limiting, and these are the 4 strategies that I'm going to talk about during this seminar. Okay, so the first thing that we did was go to Indonesia, and this was actually much more difficult than I expected. Can you see my arrow if I point here? Here's Jakarta. So Australia is somewhere down here in the right-hand corner. You fly from Jakarta to Makassar. This is a couple of hours flight, and then a very short flight, but very frightening, in a small propeller plane to our field site, Lake Methane, which is here on Sulawesi Island. And one of the things that jumps out at you when you're in this airplane flying over is that in the places where they've mined, the soil is bright, bright red. And that tells us that there's a lot of iron in these sediments and soils here. And in Lake Methane, there's also a lot of iron as a result of that in the water. So it's a ferruginous lake, it's stratified, and it's very isolated, which makes it a really interesting field site for a number of different kinds of scientists. The geochemists who have been working here for a long time have been studying it because it is so high in iron that it's a good analog for the Archean Ocean, which is thought to have been high in iron rather than sulfate like the modern ocean. It's stratified, so it's about 600 meters deep, and the bottom 500 meters are permanently anoxic. And so in those bottom 500 meters, you have a lot of iron too. There's almost no measurable sulfide. And in the surface water, as a consequence of the high iron concentration, there's very little phosphate. What happens is that the iron 3 in the surface water is not present as soluble iron 3. It's present as nanoparticulate iron oxyhydroxides. And those nanoparticulates absorb all the phosphate. And so the phosphorus concentration, the soluble phosphorus concentration in the surface water, actually can't be measured. People have been working here for a long time and have never gotten an accurate measurement of how much phosphorus is there. It's also really isolated. It got closed off from the ocean between 1 and 4 million years ago. And as a consequence of that, the fish populations are actually endemic. We have species of fish in this lake that have not been seen in any other places. And so biologists have been working in this lake for many years, looking at evolutionary biology of things like fish and shrimp. So it's a really interesting field site for those reasons, but I've been interested in the physiological consequences of very low phosphorus and how you work to get around them. Among the reasons that the lake is low nutrient is that it has very few inputs. There are 3 tributaries that look like this. They're very small streams. They're about 18 inches deep. And there's really not much coming in through those. There are a couple of towns around the edges of the lake. So this is Sorako, the town where we were staying. And they do just pretty much dump their waste directly into the lake. So there are some waste inputs along the edges in a couple of places, but they're very small compared to the total area of the lake. And then there's rain input. So during the rainy season, rain comes in. But there's not a lot of ways for nutrients, nitrogen, phosphorus, organic carbon, to get into the lake. So it's pretty nutrient limited in all senses. The structure, as I said, is kind of unusual. It's 600 meters deep at the deepest point, and the bottom 500 meters is permanently anoxic. This is just a small model of the iron cycle that Sean Crow and his collaborators put together several years ago, showing that iron is mostly coming in from the sediments. It can be oxidized as it moves through the chemocline here, which is between 100 and 120 meters pretty stably. There are pretty steep chemical gradients, so this red line here is oxygen, and you can see that all of the oxygen is gone at around 120 meters. The green diamonds here are sulfate reduction rates. There's a spike in sulfate reduction rate right around 120 meters, but there's very little sulfate reduction anywhere else because there's very little sulfur altogether. And then the blue circles here are iron 2. The chemistry above the chemocline is also not totally normal. There are high levels of other metals, including chromium, nickel, and cobalt in the surface water. And the phosphorus, like I said, is below the limit of detection. And the most recent attempt that we made to measure the phosphorus concentration had a limit of detection of 15 nanomolar. It was less than that. So it's really very, very phosphorus limited. We also know that the organisms in the lake are phosphorus limited, right? Because the question is not whether or not we can see it, but whether or not the organisms there need more than they can get. And what we did, and by we I mean Sean Crow and Carrie Ann Jones, not me, was some bottle experiments in the field where they filled up some bottles with surface water and then added phosphate to half of those bottles and measured carbon fixation rates. And what they saw was that in the bottles where phosphate had been added, the carbon fixation rate increased by about 30%. So we know that those cells are phosphorus limited and that as soon as they get more, they increase their activity right away. So if you think about how bacteria use phosphorus and where they employ it, there are 4 major pools of phosphorus in the cell. And the biggest by far is the RNA pool, so the ribosomes in the cell. The other big pools are, of course, the phospholipids in the membrane, the DNA, so the genome, and then there are smaller pools of free nucleotides. And if you're living with very, very limited phosphorus, you're going to have to reduce your demand for phosphorus one way or another. So the really big question that we started to address in my lab was, can the sizes of any of these pools be reduced, and how much of a difference does that make to how they're growing? And we can start to make some guesses about how cells might be doing this using metagenomic analysis, right? So cultivation-independent analysis. But we really need to isolate some bacteria and do some lab experiments to answer those questions accurately. So we started with a metagenomic analysis of water that had been collected from the surface, and we first just took a look at what kinds of bacteria are there. We had hypothesized, kind of based on those, the experience of those fish biologists, that there might be an unusual microbial population in the surface water, that if you have such a nutrient-limited system, it may have selected for some organisms that are really specialized for life under those conditions, and so we might have a population that would be dominated by organisms specialized to fit in that niche. But what we see is a fairly normal-looking assemblage of microbes. We see very few cyanobacteria, which I circle here. They're about 10% of the population. So it doesn't look like photosynthesis, at least at 10 meters. There is an oxygen maximum a little bit deeper, around 40 meters, where they may be more abundant. In actual fact, the biggest group in the surface water seems to be actinobacteria, so gram-positive bacteria. But there's really no individual taxon that dominates this community. Based on further analysis of that metagenome, we did look for phosphorus uptake and utilization genes to get us started, to help us decide what experiments to do first. So we did see both PST and PIT transport systems. The PST is the high affinity uptake system, and PIT is the low affinity system. And the PST is much, much more abundant. So it seems clear that these organisms are using a high affinity phosphate transporter, which is what you would expect in a phosphate-limited systems. We see several different types of alkaline phosphatases. Most of them are Pho-X, and this is similar to what's been observed in marine systems as well. But we do see several kinds. We also see polyphosphate kinases. So these are genes that are responsible for polyphosphate storage in the cell, and several different pathways for phosphonate utilization. So these are compounds that have direct carbon to phosphorus bonds rather than carbon-oxygen phosphorus bonds that you see in phosphate esters. So based on that data, we hypothesized that any organisms in this lake should be able to consume several different kinds of phosphorus. They, of course, should be able to use normal phosphate, so inorganic phosphate in a +5 oxidation state. And given the number of phosphatases we saw, they should also be able to cleave and then consume phosphate esters, so organic phosphate in a +5 oxidation state. We also hypothesized that they can use phosphonates, which are organic compounds with a +3 oxidation state, and phosphite, which is an inorganic phosphorus compound with a +3 oxidation state. And because we think that a lot of the phosphorus in Lake Metano is going to be in the insoluble pool, in either biomass or associated with minerals, we thought that those two should also be included in the potential sources of phosphorus that these cells can use. So my graduate student, Meng-Yin Yao, isolated several different bacteria, heterotrophic bacteria from Lake Metano, that we could use as model systems to confirm the predictions that we made based on field observations and the metagenomic data analysis. These are just pictures of a few. Since I'm pre-tenure, we picked out cells that grow fairly fast in the lab. They all grow really nicely on plates. And for the most part, they grow at a reasonably fast growth rate. The 6 strains that we picked for further analysis are not hugely diverse, but they are representative of the other things that we see in that surface water. Several are gamma proteobacteria, Acinetobacter species, and Pantoea. We have several isolates from the Rhizobium group and one from the Methylobacterium group. And we also have 1 gram-positive isolate, LM2, which is a member of the Actinobacteria. So those are the group that's most common in the metagenome. And of course, we grew those first just with abundant phosphate. We gave them 0.2 millimolar potassium phosphate and said, how fast do you guys grow? And you can see from this growth curve that they all grow fairly quickly. They reach stationary phase within 24 hours. And then they have a really long, really stable stationary phase. With one exception. There's one strain that instead of growing really fast in the first 24 hours and then stopping, grows really slowly for several days. And it will eventually reach stationary phase, but it takes a very long time to get there. And so this told us that there are multiple growth strategies in use by the bacteria in Lake Metano. I'm going to come back to this, so don't forget it. So to test this diversification hypothesis, then, these strains should be able to use many different sources of phosphate. We just grew them on different sources of phosphate. Each of them was provided at 0.2 millimolar in the medium, so plenty, and there was also abundant carbon and nitrogen in the medium. We didn't want them to be limited for anything except phosphorus. And these are just plots of the growth rate. What you can see here is that almost all of the strains can use all of those phosphorus sources. They have all diversified their ability to collect phosphorus from as many sources as possible. So they grow really nicely on phosphate, which is the dark blue bars, and on phosphate esters, which is the light blue bars. They can actually all use phosphite, and that kind of surprised me a little bit. Some of them grow more slowly on phosphate, but they do grow on it. And most of them can also use phosphonates, so those in the +3 oxidation state, organic. All but 2 can use that. And then all of them can use mineral-associated HFOP, which is an iron oxyhydroxide that we co-precipitated with phosphorus. So what this tells us is that those growth rates are pretty similar, and they can all use those different sources. I'm going to come back to most of these in a little bit more detail in a few minutes. Okay, so in terms of phosphate esters, we have not done a lot of experiments yet with those phosphatases, but we're starting to do that now. What has been observed in the past by Carey and Jones is that Lake Methane has really, really high alkaline phosphatase activity. So if you measure the alkaline phosphatase activity in the lake, it's about 1.8 millimoles per liter per hour. And normal for freshwater lakes is about 0.4. So it's more than 4 times higher than what we ordinarily see in lakes. And we really don't know if that's just because all of the bacteria in the lake are producing alkaline phosphatases like crazy because they're so desperate, or because those proteins are just really efficient. So I have a student now who's working on purifying those proteins from some of our isolates so that we can start to answer that question. And we have shown that our isolates have Pho-D, Pho-X, or Pho-A, and actually that one isolate has 2 of them. So our model systems are pretty suitable for going a little further into this analysis. One of the questions that's been really interesting is this question about phosphonates. I had thought that phosphonates were not that common, but it turns out that they're a major part of the organic phosphorus pool in a lot of environments, including the open ocean and in a lot of freshwater lakes. And they're these organic compounds that have a direct carbon to phosphorus bond, so this bond right here. And if you break the bond between a phosphonate and a methylphosphonate, so phosphonate with just a methyl group attached, then you release methane as a byproduct. And one of the things that has been observed about Lake Methane in the past is that the surface water is supersaturated with respect to methane. So this is one potential explanation for how much methane is there in the surface water. We looked in the metagenome to see whether or not it was possible that methane was being produced in situ via this kind of pathway. And what we see is that all of the genes that are necessary for transporting methylphosphonate into the cell and then breaking that carbon-phosphorus bond and releasing methane are present in the Lake Methane metagenome at a higher number than you would expect So this is the odds ratio, and the odds ratio is just a ratio of the number of genes that we observe in the metagenome compared to in the KEGG database. If the odds ratio is greater than 1, it means that those genes are enriched in our metagenome data set. And all of these genes have an odds ratio that's greater than 1. So that tells us that a lot of the organisms in our data set, more than we would expect, have these genes. So the first question we had about this was, well, can they use methylphosphonate as a phosphorus source? So we provided our cells with either with potassium phosphate or with methylphosphonate as a phosphorus source, and then measured growth. Only 4 of the strains can use methylphosphonate as a phosphorus source. But of those 4, they grow just as fast using phosphate or methylphosphonate as a phosphorus source. So that's all I wanted to show you with this figure, is that they grow just as fast on either phosphorus source. The second question, of course, is then, well, do they produce methane from methylphosphonate? And in fact, they do. All 4 of them do. And in the absence of methylphosphonate, they don't produce methane. So the methane is coming from the methylphosphonate utilization and not from some other cellular process. If we add phosphate to the cultures that are being grown with methylphosphonate, what we see is a reduction in the amount of methane produced. So 3 of these strains, the methane production goes down by 40 to 50%. In one strain, it's actually completely repressed. So that methane release from methylphosphonate is a consequence of phosphorus acquisition from this compound and not from some other way of using it. And this may help us to explain the quantity of methane that is present in Lake Methane. So there's a lot of methane production in the bottom waters here, right? This is all anoxic bottom water, and there's really active methanogenesis down here. And of course, some of it does bubble up through the chemocline, but a lot of it is oxidized in the chemocline between 100 and 200 meters. You can see that here where there's this rapid decrease. But even here in the surface water, the methane concentration is about 3 micromolar, which is much, much higher than what you would expect for it to be in equilibrium with the atmosphere. So in actual fact, the oligotrophic status of this lake may enhance greenhouse gas emissions, right? These organisms are so desperate for phosphorus that they're consuming small organic molecules and releasing methane as a byproduct. And they wouldn't be doing that if they weren't so phosphorus limited. Okay. The last thing I wanted to, or the last phosphorus source that I wanted to talk about here is getting phosphorus from minerals. So I had said that the iron oxyhydroxides in the lake sorb phosphorus. And so the question we had was, is that phosphorus still bioavailable even though we can't measure it in solution? So my graduate student, Mengyan Yao, co-precipitated iron oxyhydroxide with phosphate, showed that it does not dissolve over a week of incubation. That's what this abiotic control is in solution. And then provided that as a phosphorus source in culture to all of our different isolates. We also used E. coli as a negative control to see what would happen if you just have normal bacterial metabolism going on in those tubes. Will they also release phosphorus from those minerals? And because there's a because metabolism lowers the pH, you can see that here, the pH goes down to about 4. They do dissolve those minerals to some degree. And so the degree of dissolution from most of these isolates is pretty similar to what you see with E. coli. So it's really just a product of normal bacterial metabolism, but it does make that apparently unavailable insoluble phosphorus available to microbes. But one of our strains, LMY, actually actively releases phosphorus from those minerals. So you can see that the concentration of phosphorus is much higher after incubation, and iron is much, much higher. So a lot of the phosphorus that they release is taken up into the cells, but the iron is not, so that all stays in solution. We did add additional phosphorus to this strain to see if that would repress the activity, and it doesn't have any effect. You can see that there's still a lot of phosphorus and a lot of iron being released, and the pH is still quite low. But it means that that pool of insoluble phosphorus is bioavailable. Okay, so what we can conclude from this set of experiments is that there's a really big variety of phosphorus compounds, both organic and inorganic, and in the +5 and +3 oxidation states that are bioavailable to the organisms that we isolated from this lake. And in fact, all of the ones that we isolated can use almost all of these phosphorus sources. The other experiment that we did was to try to grow the cells without phosphate, right? So we just didn't give them any additional phosphate. There's some phosphorus that will go into the medium just as a contaminant from other reagents that go in there. But we couldn't detect it. So the limit of detection for phosphorus in our lab is about 3 micromolar. We couldn't detect it, so the concentration of phosphorus in these experiments was less than 3 micromolar. And what you can see is that all of these cells do still grow. They grow very slowly, and they grow only a little bit, so they don't reach very high cell density, but they do grow. And in the absence of phosphorus, diversification, or being able to acquire phosphorus from different sources, won't help them, right? So what makes this growth possible? And to answer that question, we have to go back to this list of possible ways to enable growth in a resource-limited system. And that's going to, for that, we're going to have to turn to these alternatives, right? Substitute for more abundant resources, reduce the demand, and increase efficiency. So what are the phosphorus pools that we can think about replacing phosphorus in? It seems like the pool that would be where it would be easiest to replace phosphate is the lipid pool. And so to answer that question, we collaborated with Kai Hinrichs and his student Felix Elling. We grew the cells with and without phosphate and then sent them off for analysis of the lipids. And what we see is really striking. You see that in the presence of phosphate, all of the cells produce almost entirely phospholipids. And in the absence of phosphate, none of them make phospholipids. They all start instead making aminolipids and glycolipids. And this was really surprising. If we look a little bit more closely at what kinds of lipids they're replaced with, you can see that they all make several different kinds of phospholipids in the presence of phosphate. And in the absence of phosphate, they go down to just 1 or 2 kinds, and instead are making aminolipids. These DGTS head groups are betaine head groups, and the OL is ornithine lipids. So there's some diversity in the kinds of lipids that they make, but they replace the phospholipids completely. These are glycolipids, and you can see that it's our actinobacterium that primarily makes glycolipids, but all of the other isolates make some. One of the things that we thought was interesting was that there were no sulfolipids detected. We had expected that we might see some, because that's been observed in, especially in autotrophic bacteria in phosphate-limited environments, that they produce sulfolipids. We didn't see any sulfolipids here in these analyses, and we also didn't see any of the genes required for sulfolipid biosynthesis in the metagenomic data set. We do see some of the genes for production of aminolipids and glycolipids. There was something else I wanted to say about this slide. Okay. So we can replace this close to 100%, which does reduce the demand of the cell, and it reduces the size of the phosphorus pool in the lipids. But what other pools can we reduce the size of? The biggest pool by far in this of phosphorus in the cell is the ribosomal RNA pool. So can the cells get by with less RNA? The DNA pool is also an obvious place. This is not a huge, it's not the biggest pool of phosphorus in the cell. And it's been observed in the literature that bacteria in resource-limited systems do tend to have smaller genomes. We have not looked into that yet. But to answer the question about our RNA, we collaborated with colleagues here at UD in the chemical engineering department, Maciek and his student Chris Long. And we did the same thing. We grew the cells with and without phosphate and asked the question, how much RNA do they have? And what we see is that in some cells, there was no change in the amount of RNA present. So this one and this one really don't make any less RNA. But in some cases, the RNA content was reduced by 80%. So this is a huge change in the amount of phosphorus that the cell needs if they reduce the RNA pool by 80%. So what does this mean for efficiency? Since RNA is the largest pool of phosphorus in the cell, especially in phosphate-limited environments, there were some papers that came out in the early 2000s that said that in bacteria in phosphate-limited lakes, up to 97% of the phosphorus in the cell was in that RNA pool. So if we reduce that RNA pool to 20% of its original size, we really made a big difference in how much phosphorus those cells need. And then further reductions in P demand by replacing phospholipids means that that P demand is dramatically reduced on the whole. So what we've shown so far is that they've diversified phosphorus acquisition, they've substituted for more abundant resources in the lipid pool, and they can reduce their demand by shrinking the RNA pool by a lot. And I have one more thing to say about efficiency, and then I think I'm done, and I can show you pictures of the field site, which is that these two growth strategies that I mentioned at the very beginning also create different kinds of niches for different organisms. So most of the cells that we isolated are what we call copiotrophs. They grow really fast for a very short time, and then they have a long, stable, stationary phase, right? So when resources are abundant, they take advantage of it right away, and then they can hang out for a while and wait for things to get better again. This one slow-growing LMP strain is what's called an oligotroph. So it grows really slowly, but for a very extended period of time. And this one consistently reached the highest cell densities of all of the strains that we were working with. This one is also capable of polyphosphate storage inside the cell. So if there's available phosphate, it can store it as granules inside the cell and then use them later. So we have two very different growth strategies that allow cells to inhabit niches that are different, not just in space and not just in terms of what kinds of resources they can utilize, but also in time, right? These ones will grow very quickly after some kind of nutrient input, and these ones will take advantage of those nutrients, but on a longer time frame, and they can save some of them for later. Okay. So what this tells us is that this blue water that appears to be empty has a cell density of about 10 to the 5 to 10 to the 6 cells per milliliter. So it's comparable to what you see in the open ocean. And that blue water is full of niches in both space and time for efficient and enterprising microbes to occupy. I think I talked really fast. And what we can say about this is that bacteria in Lake Matano do grow pretty well in phosphorus-starved conditions. They can use a variety of different phosphorus sources, and some of these the consequences of using these diverse P sources, it has ecological consequences, right? So we see that methane can be released from some of them. They also can replace their phospholipids almost completely with amino and glycolipids. They reduce the size of the RNA pool pretty dramatically, and this leads to a variable carbon to phosphorus ratio in the cells. So we make predictions about how well cells grow based on what we know about how much of those different nutrients they need. If what they need is significantly less than what we had predicted, so less than the sort of normal CP ratio, then our predictions may be off, and they may be off by a lot. What we're working on now and in the future is biochemical analyses of some of the alkaline phosphatases. We're looking at gene expression analysis of the genes for methylphosphonate degradation in our isolates, and looking for phosphate utilization pathways in the metagenome. We're also thinking about the biophysical effects of changes in the lipid composition, right? So phosphate is a negatively charged compound, and phospholipids in general will impart a negative charge to the cell. Glycolipids will do the same, but aminolipids aren't necessarily negatively charged like that, and so there can be some changes to cell structure and charge that would be related to those changes in lipid composition, and I think that would be really interesting. The fieldwork was made possible by Anchor. We have to thank them a lot. And also by a really huge team of people. The people here at UD who have been working on this project are primarily my graduate student, Mengyan Yao, and an undergraduate, Diana Kitt, but also my former postdoc, Jessica Keffer, and we had help from Tom Hansen, Chris Long, and Maciek Antoniewicz. The fieldwork was done primarily by Sean Crow and Carrie Ann Jones. Sulung Nomosatrio, Cynthia, and Tri are our collaborators in Indonesia. And Jens Kallmeyer, Andre Fries, and Celine Michaels were also there on that field team. The lipid work was and his student, Felix Elling, and the metagenomic sequencing was done in Ed DeLong's lab with Rachel Berry. So I do have to thank all of those people for their help with this work. And since I have a couple minutes left, I'll say that this was our collection system in the field. This is Andre and Sulung, and we dropped Niskin bottles over the side. We had this winch and a pulley system here, and the Niskin bottles had to be winched down and then back up every time. So between 100 and 600 meters, up and down by hand, we all took turns. The research vessel was this boat, which was actually quite big and pretty comfortable. And our research, oh no, I guess I don't have a picture of the research house, but here's the CTD that we were using to find the bottom and to identify where the chemocline was. So this is 150 and 160 meters, then we were close to the chemocline here in this picture. I think with that, I have no more slides, and I'm happy to take any questions. It's weird not to hear people clapping. You guys don't have to clap. I have a studio audience.
All right. Well, if anyone inside the Hangout would like to unmute themselves and ask a question, feel free to start doing so. And if anyone watching online would like to ask a question through Twitter, use the hashtag #USeminar or tweet it to microseminar. And Laura, do you want to unmute, or should I ask your question? Laura already tweeted.
No, I can unmute if you can hear me.
Yeah, we can hear you. Go for it.
Yeah, I fixed it. Okay. So my question was, you're seeing this shift in the phospholipid shift to glycolipids and aminolipids, and I was wondering if you see that in the lake samples in that low phosphorus environment.
So I'm not sure how to answer this question because I know the answer, but it's not published. How do we feel about that? Because it's not my work.
Right. Okay. I think then, like, you don't have to say anything, but you can say you've looked at it and it's interesting.
So I'll say that the results are similar.
Okay.
And it surprised me. Yeah. The results are similar. We have looked at it.
Okay.
But I have personally not, and it's not mine, so I hesitate.
Yeah, no, that's fair.
So I have a question.
Yeah.
So you were drawing comparisons to the open ocean, right? And so a lot of what you've shown is very reminiscent of Sargasso Sea SAR11 type activity, right, with the methylphosphonates. But yet you originally had shown that the community composition of the lake isn't drastically abnormal.
Nope.
And so it's impressive that, like, in 7 isolates, you've got this oligotroph along with the copiotrophs. But in the metagenome, I wonder, is there any sort of bulk parameter in the metagenome that might give you an idea if these are all more oligotrophic actinos and typical freshwater lake inhabitants, or do you see plenty of copiotroph markers also?
So the short answer to that question is that I really don't know. The long answer is that there are some things in that metagenome that remind us of the oceans. So, for example, the cyanobacteria are Prochlorococcus type cyanobacteria. There was something else that was marine-like that I can't remember. But for the most part, that community on the whole looks like a freshwater lake. What people have been finding, especially in recent years, is that oligotrophic freshwaters and oligotrophic ocean waters are similar in a lot of ways. So, for example, this methylphosphonate thing that we talked about has been seen both in freshwater and in ocean waters, right? It results in release of methane as long as they're using it as a phosphorus source. It's possible that in more carbon-limited systems that methane's not released or it's consumed rapidly because other things will use it as a carbon source. We haven't seen that with our isolates, but we also have only isolated a very small subset of the kinds of things that are available, right? So one of the things I'm hoping is that we can use that metagenomic data to improve our cultivation strategies in the future. And maybe next year, when the pressure is a little bit different, we can isolate things that grow more slowly, because I think that they might be ultimately more physiologically interesting. Yeah.
Any other questions?
Yeah, I have one. So this is my own ignorance, and I had to leave during the middle to my dumpster came. I had to show them where to come put it. So you showed that in the end that they changed their RNA pools based on the amount of phosphate there. But what does that mean? Are they differentially growing? Is that just kind of a storage? There's more RNA in one cell versus one type of cell versus the others. Like, is there a functional consequence of that change in RNA pool, or is it just the way it is?
So both of those things, they do change, and the ones that reduce the size of the RNA pool are the ones with the biggest change in growth rate under those two different conditions. So I think it's clear that when you have fewer ribosomes, you can't do as much, right? And you can't grow as fast, and that makes a lot of sense. But they are all still growing. The only one, I may not be able to show you my slides again, but let me at least look at the data so I'm sure I'm not lying to you. The only one that doesn't grow without phosphorus is one, two. Okay, let me try to share my slides again. This may or may not work. We had some technical difficulties earlier. Okay, can you see the slide?
Yep.
So the only one that doesn't grow when we take away phosphorus is this one. And this is also one of the ones that doesn't change the size of its RNA pool. So it clearly just needs more phosphorus for normal growth than it can get under phosphorus-limited conditions. So I think that makes sense. Stop sharing. So yeah, the physiological consequence of a reduced RNA pool is slower growth. We haven't looked at any of the other potential consequences. You know, is there a change in translation rates or transcription rates or anything else? But on a bulk scale, that change in RNA content changes the growth rate.
So we just had a question come in online, and it was asking, have you analyzed which kinds of RNA are being decreased? So that kind of goes off that last question, right? And that, so you're saying it's probably an overall.
I think it's overall. I mean, it's clear that, I mean, we know, right, that the biggest pool of RNA in the cell, if we're just looking at RNA, is ribosomal. And so a change in the rate of transcription wouldn't cause an 80% reduction in RNA content, right? Unless you're already starting with a really, really low ribosome number of ribosomes, which is possible, but we haven't looked at it. But I think it's unlikely that. I think it's likely that there's a change in transcription rate, so there's a change in mRNA level, but I don't think it would cause the big changes that we see overall. Does that make sense? Mm-hmm. I can see all your heads now, so if you nod, I can see whether or not this is making sense to you. Okay.
I do have a question.
Sure.
Hi, Julie. So sorry I got in late, so sorry about that if you talked about it already. This is Alex talking. Have you tried to do some stoichiometry for all those isolates? And I guess my question is, if you did, how fast did it change? And do you think we could use it as a tracer for phosphate starvation in other types of systems?
Yeah, so this is a really interesting question. We haven't, but I've been thinking about doing it and how to do it. But I don't know if we could use it as a tracer for phosphorus starvation, because that C to P ratio is so variable in different systems, right? It's in Lake Metano, we think that it's greater than 1,000, but in a lot of freshwater lakes, it's in the hundreds, and in other places, it's in the tens. So it does vary dramatically depending on what kind of bacteria you have. Let me hold my fingers up so you can count. What kind of bacteria you have, what the conditions are. So it can change in the same bacteria just on the conditions. And what the ratios of the different kinds you have are. Does that make sense? So I'm not sure that it would be a good tracer, but I am really curious to see how the C to P ratio changes in our particular isolates under different conditions and over time.
Good. And so I missed this at the beginning, but did you see changes of? It's a very deep lake, and so did you see changes in the presence of those isolates over depth, or how is this phosphate concentration structured?
So the phosphate concentration, I can show you. We don't know how the microbial population or community composition changes over time, because we've only done metagenomic analysis from 10 meters and 120 meters.
Okay.
So they're very different, but 120 meters is right in the middle of the chemocline, so it's right where there's at that oxic-anoxic transition zone.
Okay.
So here's the phosphorus concentration in the lake. It's abundant below the chemocline, right? So where the iron is reduced and no longer sorbing the phosphorus, it's abundantly available. But at the chemocline, where you start seeing formation of iron oxyhydroxides and sorption of the phosphate from solution, the phosphorus all goes away. And then it's basically not measurable above the chemocline.
Okay.
Does that answer your question about the structure?
Yeah. And so those isolates those strains were isolated from that first 100 meters?
From the top 10 meters.
Okay.
I think.
Thank you. Sorry, I missed that at the beginning. Thanks.
No problem. No problem.
Okay. All right. It looks like questions have slowed down. So thanks to her starring in Micro Seminar, Julie has been peer pressured into joining Twitter finally.
Yeah.
So you can tweet her directly at Civil Micro, and she will happily answer any questions. And thanks to everyone for participating today. Next month's seminar will be John Klassen from UConn. And we will see you all next month. If anyone is interested in giving a seminar, please feel free to get in contact with us, and we will sign you up.
Thanks a lot, Jen.
Thanks.